Namaste students, Pushpa Vidya, I JP and Dr. Rajan Nicola, Assistant Professor at the Department of University of Jaipur. In this session, we are going to discuss about population variation, that is selection. Okay. So first of all, one of the common misconceptions in that the organism evolved during their lifespan. So the natural selection acts on the individual, but only the population evolves. Okay. So here, for example, a population of in a medium or a ground range, okay, like during drought, large big birds are more likely to track the large seeds and survive. So here, here we can see different populations or different during the drought, like finch populations evolved from by the natural selections. The microevolution is a change in allele frequency in the population over generations. So three mechanical mechanisms cause the allele frequency change, that is the natural selection, genetic drift, and the gene flow. Okay. So our natural selection causes an adaptive evolution, like the conception or a genetic variations make or the evolution possible. Okay. So the variation in the heritable traits or a prerequisite for evolution. So Mendel's work on a pea plant provided evidences of the discrete heritable unit or the genes. Okay. So the phenotype, when we see the genetic variations, the phenotypic variations often reflects the genetic variations or the genetic variations among the individual is caused by the differences in the gene or other DNA sequences. Okay. So some phenotypic differences are due to the differences in a single gene and are classified or either or basis, or the other phenotypic differences are due to the influence of many genes. Okay, vary in a relation along the continuum. Here, the genetic variation can be measured as a whole level as genetic variability of the genetic variability can be quantified as the average percentage of the loci are heterozygous. Okay. So genetic variation can be measured at the molecular level of a DNA and the nucleotide. Or the nucleotide variations rarely result in the phenotype of the variation. The phenotype is the product of the herited genotype of the environmental influence. The natural selection can only act as a phenotypic variation that has the genetic components. So the different like sources of the genetic variations are new genes and allele can be raised by the mutations of the gene. How a new formation of new allele takes place. So the mutation is a change. Okay, mutation is a change in the nucleotide sequence of the DNA. Okay. So the only mutation in the cells that produce gametes can be passed to the offspring. And the point mutation is a change in one base in the gene. So here the effect of the mutation can vary. The mutations is non-coding region of a DNA are often harmless. Okay? So the mutation to a gene can be neutral because of the uh, genetic its genetic code. So here there is an effect in the point mutation, which can vary in a like mutation that alters the phenotype of it, and often the um, harmful mutations which results in the change in the protein molecules. So it can cause the same benefits. So alternating the gene and the number of position, that means the chromosomal mutation that deletes or restructures or rearrange many loci are typically harmful. Okay. So here duplication of the small pieces of the DNA increases or the genome size is usually less or harmful. So the duplicated genes can take on new functions by further mutations. Okay, so the ancestral order or detecting the gene has been duplicated many times. Okay, the human has 350 functional copies of the gene, or mice have thousands. Okay. The mutation rate are lower in the animals and plants. So the average is about one mutation in every 1 lakh gene per generation. The mutation rate is often low in prokaryotic and high in virus. Such short gene generation time allows to accumulate rapidly in the prokaryotes and the viruses. Okay. So when we see the sexual reproduction in organism that reproduces sexually, most genetic variations results from the recombination of the alleles. Sexual reproduction can shuffle existing the alleles into a new combinations, and there can be mechanisms. Okay, so the crossing over, independent assortment, and the fertilization. Okay. So the Hartley-Weinberg equation use the test where whether the population is evolving. That means the first step in the testing whether the evolution is occurring in a population is to clarify what we mean by a population. Okay. So here the population is a localized group of individuals capable of interbreeding and producing a fertile offspring. Okay. That means a gene pool consists of all the genes for all loci in a population. An allele for a particular locus is fixed if all individuals of a population are homozygous for the same allele. Okay. Here, the frequency of the allele in a population can be calculated for a diploid organism. The total number of alleles at least the locus is the total number of individual times. That means the total time of the dominant alleles at the locus is 2 alleles for each homologous dominance. Okay. So here the individual is one for each homologous heterozygous individual. That means the same locus appears for the recessive gene here. Okay. So by convention, if there are 2 alleles at a locus and P and Q, so used to represent their frequency. Okay. So the frequency of the allele in a population will be add up to 1. Okay. That means P plus Q is equal to 1. Okay. For example, if we consider the population of a flower here. So considering the flower population, we are considering it. Let's say so here you can see. Let me share the slide. So here in this image you can see the image. So now you can see here the two flowers has been taken, which is like, for example, here the consider the population of a white flower. Okay, that is incompletely dominant for the color. Okay, here we are taking the incompletely dominant one. It is incompletely dominant for the color. Okay. That means here the three flowers of capital R, three with capital R and capital R. Red is the dominant color with C with here white and here with the white and red. That is the pink one. Okay. Here the 320 red flowers were obtained. 160 flowers will be like this and 20. That means the here the ratio will be the calculation of the number of the copied alleles will be this much and this much. So the calculation of the frequency of each allele is minus p is equal to frequency of the people to the power r. That means 800 by 800 plus 200. That is the 80%. That means q minus q is equal to 1 minus c. That means the 20%. So the terms are of the allele is always 1. Okay. So here the Hartley-Weinberg principle describes the populations that are not evolving If the population do not meet the criteria of the Hartley-Weinberg principle, it can be concluded as the population is rebounding and forming an equilibrium. That means that state that the frequency of the alleles and the genotype in a population remains constant for the generation to generate. Here from one generation to another generation in a given population where the genotype contributes to the generation and the next generation. Mendelian inheritance preserves genetic variation in a population. The Hartley-Weinberg equilibrium describes the constant frequency of an allele in such a gene pool. Okay. So here the p square p plus 2 qp plus q square is equal to 1. That means the expected frequency of a genotype plus expected frequency of a genotype plus expected frequency of a genotype is always equal to 1. Okay? So the condition of the theorem describes the hypothetical population that is not evolving. It is a in a real population, alleles in the genotype frequency do change over a time. Okay? So the 5 conditions of non-evolving populations are rarely made in nature. That is no mutation, random mutating, no natural selection, extremely large population, and no gene flow. That means the natural population can evolve at some locale while being partly involved equilibrium at other loci. Okay? So the population of the evolved slowly enough that the evolution can't be detected. So applying this, the phenylketonuria is of the locus. The mutation there is rate is rare. Okay? So the made selection with respect to that, not at the individual or carrier for the phenylketonuria. So the natural selection can only be rare or homologous. Okay? The population is large or mitigation is thank you. This session is powered by 2.0 Vidyapet Women's University, Jaipur. If you have